Hello. Hello, everybody. I think you can see me. Yes. So, earlier, if you were here for the data science and coffee and dogs, um, we were trying to fix my car. We didn't finish it, but we did get one of the parts off, which is great. So now all we have to do is figure out how to put the front axle in. But now we're going to go into our data science. We are working on currently an autistic database. It's not really autistic. I mean, if you're into psychology, you already know about autism. In chiropractic school, we learned about autism, the spectrum, and actually how they have to be on intermittent fasting because high glucose levels actually makes them have flare-ups. And then in this article, there's already some genes that are related to autism. I could pull up the Merck manual and then look it up, but I was really doing it more because I wanted like superhuman powers and I wanted to see what the genes are. And on NCBI, when I looked up those things like psychic, telekinesis, time travel, none of them were there, and it gave me like a forbidden page. But they did, when I looked up circadian rhythm, they did pull up the autism. So there is a study on autism, and it goes through the language type of the spectrum disorder, mild, and the savants, which are the really smart, crazy, machine-like memory, computer-like memory, where they remember and recall the most random things, like what color sweater a celebrity was wearing at an event 25.35 years ago or something like that. Because they saw it, heard it, recorded it, and they could just be eating like a sandwich. They don't even have to use neuroplasticity because they just instantly remember it. It's just part of their amygdala where they just remember things by memory. Like smell doesn't trigger it. They just, it's like they just instantly got like excitement and it triggered it and it's like there's actually a computer chip in them. So actually that's the only reason I'm really doing it, because a lot of people are apparently claiming to be on the spectrum disorder, but that's not really why I'm doing it. It's actually because it's not that I don't care about their disorder. It's just that there's so many dysfunctional people out there that like I'm not doing it for that reason. I don't attest to being able to handle people with mental problems. I actually have it in my consent form that if you do have mental problems, I am not capable. I'm not in that healthcare profession to deal with it. Okay. I can be respectful and courteous, but I mean, if I'm disrespected and offended, like, why would I want to do that? No, it's a mobile massage company, so I don't want to do that. I don't want to deal with that. Okay. If you have outbursts and crazy stuff or do offensive things, I'm not going to like it. I'm not going to be around it. It makes me uncomfortable. So no, I don't want to do that. That's not my profession. I'm a massage therapist and a data scientist. Okay. I have many hats that I wear, but a mental care worker is not one of them. I offer massage therapy, as in therapeutic relief for your soft tissue if it has aches. Okay. That's your musculoskeletal, tendinous, ligamentous structures. Right. And then I have health advice from 3 years of chiropractic school. I did 2 years of data science. I've taken the Coursera course before that and learned R version. So I stick with R. I don't want to waste time. I don't want to promote somebody else's software so that some recruiter out there can sell a software to a big company that has millions to pay on it to get all of their workers to use, because all of the new candidates or all the employment candidates in that field, because they want to do different or go on a different stream, are using that software, because I'm not using that software. If you want me to use that software, you have the company contact me, hire me, and train me and pay me while they're training me to do it, because I have proven time and again that I learn efficiently. I learn the concepts, I understand what is going on, what I don't understand, and then I learn it, fix it, and I get it right, okay? And then it's there forever, okay? And I can recall it with neuroplasticity. I'm not a savant. That's just the way my brain works, okay? And so we're going to go into this. If you don't like beer, I mean, I understand. And so this is free work. I'm not getting paid. I'm not drinking on the work. This is my hobby, okay? I have dogs. I have beer. I work, okay? So, like, if you don't like beer because you have some problem, like my dad doesn't drink, and that's cool. I'm not going to pressure him into drinking. If you want to stay here and you want to learn data science or whatever and pick it up as you go because you can, then fine. If you don't want to drink, then don't, okay? But data science can get pretty stressful. You just need to relax and chill, okay? And then we will be drinking water between each beer. This is my office. You like professional stuff? I have my chiropractic books there, and inside that black cabinet is my undergrad years of all the crazy math books and economics. Yes, you could get lost in that. And then let's go to it. All right, I'm going to share my screen. And we're just going to pick this window. And then we're just going to share it. All right, and I am going to just go to a different window. I'm hoping maybe I never noticed. I didn't really check earlier to see. I didn't even drink some of this beer yet. Okay. It is Bitburger. I get it at Trader Joe's. It doesn't leave an aftertaste. It's like the cheap version or like the real version of Stella Artois, which is a nice taste that's light and it's a lager, but it doesn't give me like this weird, filmy, like greasy taste afterwards. So that's what I drink. It's like it has an old-fashioned Regis Philbin guy on the image. Trader Joe's, $6.99 for a 4-pack. About 5 to 6% alcohol per volume. Really, you can limit yourself to 1 to 2 beers a day. Okay, you're fine. Don't even trip. So we're gonna go into the Rstudio. You can download Rstudio, but you have to download R first. You get that from the CRAN, like Cranberry Network. You can just look R statistical software. So let's go ahead and open. And I think I'm sharing. I'm not really sure what you're seeing. So, oh, yeah, you know what? Let's stop this screen share. I'm going to go ahead and, no. What is this? No, new. No, I don't want the new version. I just installed this version. All right, I have to unshare this and start sharing again. Hold on real quick. Stop sharing. Now I'm going to share. Entire screen. I'm just going to pick entire screen. Okay, yeah. We'll share that entire screen. And then I think you can see. Yes, you can see it. All right. So here we are. You get Rstudio. You're gonna have to download all these packages. It's pretty installed. All packages. So what happens is if you need any of these packages, you install them. It's usually something that they do automatically as long as you're hooked up to the internet. Oh, I hate when this cloud thing is on. Don't like it. It's on. I don't really want to. I have a Dropbox account. I don't need to. Okay. So we're just gonna run all chunks. I was working on this earlier. We can go to the site. This is GSE 15402. You can look that up. It'll take you to the gene expression NCBI, which is National Center for Bioinformatics information, or that, and then you can download the files. If you want, we can look at that site. So we'll just Control-C, go to a new page, and we will paste. And then we will see. Okay, so here we are. Gene expression profiling differentiates autism case controls and phenotypic variants of autism spectrum disorders. Expression profiling by array, microarray. There's different ways to get the gene expression data. They're basically looking at your your messenger RNA, your cellular data, your DNA, which is basically DNA double helix strands, complementary DNA copies the other strand, messenger RNA copies the complementary DNA strand into the actual DNA strand, except that U replaces T. So there are 4 nucleotides. You got adenine, thymine, guanine, cytosine in the DNA. Those are okay. Every little tiny cell, can't see it. Inside that cell is a nucleus. You got organelles outside that nucleus. That's where all the protein is made, but it has to come off of each little chromosome, tightly packed around into the nucleus. So DNA tightly packed, circles, loops around little beads called histones, and then they are wrapped around the chromosome. You got like millions of series of these nucleotides in a strand, just 4 letters for each of those nucleotides. It takes those, and those make up in sets of 3 triplets codons, 3 nucleotides, an amino acid. An amino acid, there are letters for each because there are less than the number of letters in the American alphabet or English alphabet. Yeah, and so some of those amino acids, they make the same one, but they have different nucleotides that make it. Now, amino acids are only made of RNA, which is ribonucleic acid, and that is also 4 nucleotides long. But like I said earlier, the uracil replaces the thymine. And the binding order is basically they want to use the mnemonic. I'm not going to use it though. A to T, C to G. And then whenever you have ribonucleic acid, it's A to U because there's no T, and G to C. G to C doesn't change. There's different research topics out there, but if you wanted to look up something for autism or savants, we could just look it up. We could go back to the homepage, or we can go to the Geo, and then we could just look up savant, but we already have it here, so we're just going to do that, okay? So basically, this is the information. It's a summary of the experiment, the gene expression data. They do have a article there. These are the researchers. This is a peer-reviewed thing, report. So here's the PMID. That's the article. So it was submitted 2009, but it was actually updated 2019. So basically, their article is called Gene Expression Profiling Differentiates Autism Case Controls and Phenotypic Variants of Autism Spectrum Disorders. Evidence for circadian rhythm dysfunction in severe autism. Yeah, so autistic people, they have a circadian rhythm problem. So they wouldn't make good massage therapists because they would probably like really lose it, and their rhythm would be off, and it would not be relaxing at all. I mean, I've never met a severely autistic massage therapist, but some people have speculated that other people that seemed a little off were autistic. I'm not really sure. So anyhow, they do usually need, depending on the disorder, how severe it is, they do need assisted living. So yeah. And yes, everybody knows someone can get violent. You just don't try to provoke them, and you just stay away from them. Everyone's responsible for their own actions. Like already letting a violent one out or whatnot could be like training someone with a loaded gun and then like something like that. But yeah. Anyways, we see the number. These are the samples, and then you can go into each one, and let's see if we get more information. Lymphoblastoid cell line, LCLs. It's derived from the peripheral blood mononuclear cells. So they get the LCLs from the PBMC. You'll see that often. I haven't seen that anyways. Total RNA was isolated from LCL using the trisyl isolation method according to, okay, and then you will go into manufacturing protocols. If you work in a research institute, you will probably run into these machines, and then you can do it, and it will have some help for you, and you can learn from it. But these machines are like millions of dollars, hundreds of thousands to millions of dollars, probably even hundreds of millions of dollars, and all of the materials for it. And then you have to order them from different places that make it and specialize it. There's a control process for doing all of this. As you can see, 10 micrograms of total RNA were primed with 2 microliters at random hexamer primers, 3 milligrams per milliliter at 70 degrees Celsius for 10 minutes. Then reverse transcribed at 42 degrees Celsius overnight in the presence of 400 U superscript 2 RTAs in vitro gen and 1.2 microliters of 25x amino allyl DNTB mix. Okay, so you get the idea. It's stuff that you really don't need as part of the data science project, but you know what they did. So if you do get familiar with this, you can understand where it fits when you try to compare different gene expression data and try to find error that you can fit or correct for. Usually you can't combine them. That's what they say, even though they're all, you can pretty much evaluate them based on what is overexpressed. So in cancer and basic cellular dysfunction, there is a process where the cell is supposed to be killed and destroyed and not replicated if it is dysfunctional. Okay, so in the process of DNA, if there are environmental factors that destroy it, such as radiation, viral infections, heat, extreme heat, stroke, lead poisoning, any kind of poisoning, any kind of environmental factor, sometimes genetic, that causes a disruption in the DNA. It could be one nucleotide replaced in a whole gene. It could be a few. They call those copy number variants, CNVs. So those are a gene that somehow got printed out because there's introns and exons of those nucleotides, and not every nucleotide is part of the gene. Some of them are naturally dropped anyhow. They're just filler spaces because maybe it's for the looping whenever it loops around in that sequence around histones. But there's a lot of regions of the DNA that aren't coded. And so they only have so many genes, but billions and billions of regions of the DNA. And they say that some viruses, such as Epstein-Barr virus, because I've been studying that lately, they like to live in these regions of low gene density, such as LMP1. That seems to be one that's very popular in the Epstein-Barr virus nasopharyngeal cancer. Yeah, so that one is an area where it can hide in those low density regions. I think they said chromosome 1 for that one. But then for the lymphomas, they've been saying chromosome 9. They like to hide in that area. Epstein-Barr virus also has been said to be in 90% of the population. People who get that, if they are really deficient, really stressed, their immune's not doing so great, what happens is that Epstein-Barr virus ends up taking over. It takes the DNA. It's an environmental toxin, controls the cell, starts doing some signaling, cell signaling, mayhem, disrupts things that are like the oncogenes, proto-oncogenes, so that it can cause mutations and cell dysplasia or cell hyperplasia or aplasia. Just a whole bunch of different cells that aren't supposed to be there are actually getting mutated. And it can cause tumors. It can cause Epstein-Barr virus has been connected in a long-term latent reactivation form into multiple sclerosis, as well as later types of Hodgkin's and lymphomas, large B-cell lymphomas, Burkitt's lymphoma, natural killer T-cell lymphoma. All of these are in states where the immune system is a little bit deficient. It's been weakened. Stress can do that. So normally the DNA that will get repaired and replaced by these, I guess they are like transcription factors and other contents like organelles of the cell. They will sit there. I know there's like the Ross genes and P53. They go in there and they make sure that the cell's fine, the cell's doing good, because they've got the mitosis. It's actually like the different 4 phases of cell regeneration. And at some points, they even break it down into when the cell regenerates, if it is in the actual regeneration stage: anaphase, metaphase, prophase, telophase. You can look those up if you want. YouTube has some great videos for it. I'm gonna take my first sip of beer. Okay, and so getting on with this, that was just a background of gene expression data, how this affects us. So why do they come up with genes? Why do we want to find these genes? Well, these are regular genes, these copy number variant genes. They can actually start creating those known syndromes, different gene expression, and certain genes that are not so upregulated where they're having a high expression value, because these little proteins that are in the cell, so the nucleus makes it, transcribes it in the nucleus, takes that DNA, complementary DNA of the other strand, and then it says, this is good, this is a gene we need, let's send it out, right? So then it starts translating it into the cytoplasm outside the nucleus, where a ribosome is needed, and that is basically a 2-part mechanism that creates a ribosome from other factors, other genes, the proteins inside the cell. They come together, and then these little DNA molecules, complementary DNA molecules, they are called so messenger RNA takes a hold of it. It's actually pre-messenger RNA. And so the pre-messenger RNA, it has those introns, exons, and then this is where the copy numbers variants come out. It's still the transcription phase. Transcription factors go in there like boom, boom, drop, 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 keep, right? So they get rid of, I believe it is the exons. Okay, so it's whatever doesn't sound like it should. Like exon sounds like exit, so I guess they keep the exons, and then the introns they like boot out. I don't know, whichever one. It just doesn't sound like it is something that makes sense. One of them, okay? You could look it up. It's really not that important to understand the concept. Okay. You just know that that gene has a copy number variant. It might be very similar, it might not. But if your body needs it, it needs it. So it's going to make it. And some of those proteins go into other phases. There are the shape of the protein after it's made. But okay, so we're at the pre-messenger RNA stage. Drop the introns, extrons, whatever. Keep, drop. Okay, it seems to be a good gene. Let's go. Let's send it out. Boom. That's our messenger RNA. That is when it is in. So pre-messenger RNA. Yeah. So transcribe complementary DNA. Okay, and then it makes it sends it out once the so that's pre-messenger RNA. It makes the messenger RNA from that copy. So then it goes in the cytoplasm. It is messenger RNA where it goes to the ribosome. The ribosome makes it. Ribosome is 2 parts of 2 proteins coming together. They're just like, boom, safe here. Let's do blah, 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 blah. And then they're like, we're ready. Send us that messenger RNA, right? And it is a set of triplet codons. There is a start. There is an end. And there are certain factors that are the amino acids that start it so that it can create the program, goes in, creates that amino acid, creates a protein string. That protein string is in there. That's what we're counting, that protein. But here's the thing, because of the organelles that are in the cell, we have mitochondria. Mitochondria is where we get our energy. You hear that, the ATP, where we get all the ATP from the Krebs cycle, the citrus acid cycle. That is sometimes something that is an indicator of cell death, an old cell getting destroyed. And so it will leak out mitochondrial DNA. Mitochondrial DNA is separate from the regular DNA that makes our cells. Mitochondrial DNA is a history of our ancestry, but it's also from only the mother's side because it is an organelle. It is inherited from the mother's ovum, oocyte, whenever it splits in the mitotic phase. The the fertilized one, the one that is strong, the two eggs, and then they're like, they make a cell, the cell that divides, and then there's that cytoplasm. That cytoplasm has those organelles. You inherit all of this from your mom. So sometimes if you're like a mixed person, you're mixed because you just look like it phenotypically. And then you get like a DNA background, and you're like, what the hell? Why isn't this one in there? And they're like, oh, that's right. Because it's tracing mitochondrial DNA. It's tracing your ancestry. That's why. So that's what it looks at. And it's not going to tell you your DNA, but unless you get a full DNA gene, so that's the problem with drinking beer while you do data science. So you can't really learn the concepts that well. But we do have neuroplasticity. We're not destroying our cells, okay? We're just getting rid of inflammatory factors. So here's the thing. You can get a full gene copy and learn everything about your genes and see if you have risk factors for certain diseases, because when they find these proteins that are overexpressed in certain pathologies, they take it of the person who has that pathology. They can take it at different states of that person. Maybe when they're younger, they had it and they got a disease, and then they're like, what's up with this disease? Let's compare it to their protein levels when they were healthy, or to somebody in their age group, their gender, their demographic region, like the Midwest, the South, they have to do that because of all the environmental factors that are there that people evolve and adapt to. There are certain changes in the gene, so you want to compare it as close as you can to someone who's basically the twin that doesn't have that, but you can't really get that, so you have to go with what you got. So you won't really see perfect gene expression data, but you could get an idea. So whenever you see a lot of gene, they call it up regulation, when it's overexpressed, like comparatively to, like, let's say the mean value of a group. Let's say you have 3 samples of a control, the healthy person, healthy groups of people, then you have 3 of the pathology that you're trying to look at, and then you're just like, okay, let's compare these. So we're going to get the mean of the control, every gene. It shows every gene in this gene expression profile. It's a profile. So they have different ways of taking the cellular data, gene cell data, gene expression data. You're going to have arrays. You're going to have high throughput sequencing, next generation sequencing, different kinds of ways. I've been seeing some stuff too. I've looked at some single cell RNA. The way that they look at that is a little different because they use barcodes on the actual fragmented genes. So you'll have fragmented genes and barcodes on them, and they put those together to see how many counts of a gene are in a type of media. So you can have the peripheral blood mononuclear cells, the PBMCs, the lymphoblastic cell line, LCLs. You can have the actual tumor cell tissue. You can have any kind of body tissue sample gene expression that you can basically put in an array, in a machine, and it will literally, it has a way of getting into each of those nucleotides, and then it strings them together. It sees what it is, and it just has this magnificent way of calculating everything. But you have to do it however it says to do it for that machine, because if you mess up, there's going to be error, and then there's going to be some counts that are way too high in the sample compared to others, and that might not necessarily be that person. It could just be an error in the way that they took it. So you'll see sometimes we'll have samples that are repeated once, twice, 3 times, or something. Repeat 1, 2, 3. So I did take chemistry 1A, and that's where I stopped for a prerequisite to get into chiropractic school. And it was super interesting, but super hard. Intro to chemistry was great because it went over everything. I took the genetics. That was super interesting too. And biology, very interesting. You should really get into your science courses. Okay, they really help a lot. You can also learn stuff from YouTube. You could just look at the stuff you need to learn. If you're good from learning from the book, then if you have a good book that's written in the language you speak, or even the dialect you speak, or in like how you speak, like, not like old English and layman terms that you have to look up and then you're like, oh, what does this mean? No. Well, you can learn from something like that. And then it'll it will let you understand it a little bit better. So here we're gonna now go and so what we're doing is okay, so I do fold change, okay? I do a comparison of the average value of the pathological samples over the average value, which is called the mean of the healthy samples. Both of them, the average is the mean. However, we know that the data can be skewed depending on a sample in the group or misbalanced, not balanced samples. Like you might have like 3 pathologies and only 1 healthy, or like vice versa, or maybe 30 versus like 2 or 3. Doesn't really do much, but I mean it gives you a little clue, I guess, to what could be going on. And then you can see the full change values. That's what I did for mine. And yeah, so full change is great. Tells you the average, tells you like an upregulation or downregulation. So you're gonna know if that gene, that protein was used more in this pathology, it's gonna be upregulated, or it could be downregulated. Because if it's downregulated, you could have a mechanism in there that gene is needed to protect the cells, and then it's downregulated. But this one's upregulated, so they can work in I guess what is it? Synchronizingly together. They work in sync. So you don't get rid of the downregulated ones and just take the upregulated ones. You got to look at it all. So I like to take the top 10 upregulated, top 10 downregulated. But you have to exclude all the zeros because it just does it alphabetically. So we try to get as much of something that can tell us information and doesn't. And the zeros sometimes, because they are scaled, 0.00000000001 might actually have some information, but the computer might cut it off because it's, whatever that is to the 10, like 1.0 times, I guess 1 to the negative 8 or 10 to the negative 8. So it's going to cut it off, or to the 12th or whatever. But that's why they scale it, to make it seem similar in the same window, in the same realm. If it really does have an effect, considering all the other cells and the other information there, we will probably have more information depending on how upregulated it is. Although if you have some, we just make these assumptions, and that's what it is. I made these assumptions, okay? But most of it did come from economics, econometrics, and statistical analysis, also data science, the master's science. Basically, there's algorithms. There's really not that many algorithms. You don't have to learn all of them. There's simple basic codes for R and Python. I did actually do Python in my grad school, but I actually didn't really like it because the internet wasn't that great back then. And we had to look and get frustrated and look again. And there's a little bit better now, but, you still run into errors. But sometimes, AI is really trying to win everything. So it'll see what you post up there now, and then it'll be like, I'm going to get that. I'm going to answer that. And then you'll go back the next thing. You're like, oh, look, there's an answer. So, all right. So, okay. We're just a little bit in. So now you know upregulation, downregulation. I'm using the fold changes. If you do have a median, if you do have imbalanced samples, the median is great because that's a real value. So you won't have any outliers if it's close. If the average is close to the median, that's great. But then again, it will still take the average if the median is between an even number. So it'll take the 2 numbers in the middle and average it. So if they're the same, hey, that's a pretty good reason to think that that's a good number to go by. So we just use that. So now we go into the computing part. Rstudio. I like to use the Markdown because I can go back and look at what I did. So what is this? What are you doing? New file, R Markdown. That's what I use. Has these little chunks. You got to do these little things right here. It's right above the tab on my computer. You don't use the shift sign. It's just a little tilde. Actually, it's not the tilde. It's the one that's not the tilde. Okay. I guess that's what happened when you screen share. All right. So I'm not sure how the processing is going to go with this, but let's just go ahead and run its trunk. I'm gonna run all the chunks because we're gonna see where we were at earlier. I probably won't even go anywhere. Okay, let's just go through this. I want to drink a beer. You do you. Anyways. So we took the autism data. We saw earlier how many samples. I'm using R Markdown because it is a library that will basically whenever we knit this, when it publishes it, it's going to publish as a PDF, and it's also going to ask if you want to post it to RPubs, which is where I put a lot of my work. Actually, all my artwork goes up there. And it'll give you these warnings. But see this little part right there? You can just say warning, and it'll usually pop up something. Yeah, and you just put F for false. Or you could put evaluate equals true, evaluate equals false. Include, do you want to show it or not? Same thing. You can go through all those things, and then if you run it again, that warning will disappear. But it's still running these other chunks for some reason. I think it's because the screen share is taking up a lot of my processing power. Okay. Yeah, so I actually didn't even share this with you. Oh, really sorry about that. So, and then honestly for this one, what I did was, so I downloaded these files, and it comes as these MEV files. I haven't worked with those before. I manually unzipped each one. When I unzipped each one, I put it here. Now I got each folder. But guess what? There's also a file in it with the same name. So then I went and manually for 116 of these files, just like with the unzipped one, I went to put them each in this folder because I'm not sure how I'm going to open up this MEV file. Okay, it's an old one. It's off of the Tiger one. If you go to the research article, it might say that how they did it. It will definitely let you know where this is going. Okay, introduction. Autism spectrum disorders are pervasive neurodevelopmental disorders that affect a broad spectrum of functions but are diagnosed primarily on the basis of deficits in pragmatic language and communication, impaired ability to engage in reciprocal social interactions, as well as by stereotyped and repetitive behaviors, often coupled with restricted interests. Although many genetic studies have provided evidence for high heritability, there are still no genetic markers that are unequivocally diagnostic for idiopathic, unknown autism spectrum disorder, ASD. This is in distinct contrast to the genetically defined syndromic disorders, such as Fragile X, that is another mental deficit. They call it mentally handicapped. It is based on the X chromosome. Yeah, that we learned that in chiropractic school. And oh yeah, Fragile X. So that's every time your cell replicates, you have a protecting type of barrier on the genetic string for each gene that regulates. So eventually, in those Fragile X, it's like a bomb went off on their end. So like they can only live for so long because it's only gonna develop healthy ones for so long. And then when it gets to that end, it's like it loses it. Like every time it replicates, it kind of loses something. They call it a telomere. Telomere, which is basically a strand of nucleotides that don't process anything. It's just to hold it in place for whenever it replicates. Because our body is basically programmed. We're all programmed from little biota bacteria and proteins. They were so smart, they created us. That's what they tell you in biology. It's not because we were all evolved from basically bacteria. Tuberous sclerosis, Smith-Lemli optic. I never heard of those. Rett syndrome. I'm thinking that's Tourette syndrome, in which a fraction of affected individuals are also diagnosed with ASD. The difficulty in identifying genetic variants for idiopathic ASD is often true to the heterogeneity within the ASD population, which is manifested by the broad symptomatic profile of individuals on the ASD spectrum. Another problem associated with the majority of genetic analysis is that the individual single nucleotide polymorphism, SNPs, those are whenever you, and copy number variants identifies as candidate biomarkers, even if replicated in a separate cohort, are each associated with a small percentage of the general ASD population. So they're saying they have found some, only like up to 2% of the ASD population, or the diagnosed ASD population, because you have to be diagnosed clinically. by following a questionnaire. So it's a tool they use clinically. Have shown that there are actually some genetic mutations that have caused different gene variants to be reproduced and single nucleotide polymorphisms as well, which could be just the single nucleotide that makes the copy number variant. It's different from most people in that gene. Although the majority of studies directed towards biomarker identification for ASD have focused on genetic variants, both common and rare, we hypothesize that gene expression signatures may also be informative with respect to identification of ASD. However, realizing the challenges presented by the heterogeneity of ASD, we first divided the ASD population into subgroups showing similar symptomatic profiles. So they saw these ASD people and they divided them themselves. As shown in our earlier studies, dividing the ASD population into subgroups on the basis of cluster analysis of 1, 2, 3 scores from the Autism Diagnostic Interview Revised. This is their tool they use, instrument. This is their survey, basically answering questions similar to like a concussion survey. Resulted in the identification of distinct but overlapping gene expression profiles that characterize each of the 3 ASD subgroups, analyzing comparison to non-autistic controls. In the current study, we conducted meta-analysis of our published gene expression profiles of lymphoblastoid cell lines from these 3 phenotypic subgroups of ASD to identify differentially expressed genes that were robust in separating cases from controls. Here we applied class prediction algorithms to identify and test differentially expressed genes that provide high sensitivity and specificity to separate cases and controls. A fraction of the differentially expressed genes for one of the subtypes was confirmed by high-throughput quantitative nuclease protection assays, and then validated in part with a new set of samples, demonstrating the potential of this approach for developing a biomarker screen that can detect subtypes of ASD. A biomarker screen. So maybe as early as little kids that are still developing their immune system and haven't fully developed their lymphoblastic cell line. Lymphoblastoid cell line. And materials and methods. Okay. Selection of samples. Okay. Okay. Symptoms. Cell culture. I'm just reading. Gene expression analysis using DNA microarrays. Okay. So, anyways. You saw what I did with the, oh, yeah, I didn't realize it does that. Okay, so, yeah, we'll just go ahead. I don't even know what you did. Okay, I did that, okay. Yeah, so here we have the series information, which is basically the text file that it comes with. So back on that site, if you scroll to the end of it, I could have showed you where this comes from, and then it's just a text file, but this puts it into perspective, and it's usually the same repeated stuff for each sample. So as a text file, it's messy, but you can still read it to know where you need to divide the table at. And here, I had to read in only 35 rows because when I got to the 36th row, I was saying it didn't fit this right here, and that could be for some symbol that it has. I'm not sure. Actually, that's pretty much why. So you have to mess around with it until it works. There are programs out there that do that. But right now when I do this, it's like just finding the genes that can basically show if it's up or down regulated. I'm just doing this kind of like, I just keep doing it, and that's what it is. And sometimes I learn new things and I have to learn a new program to access certain information. But you can just hover over it and it'll tell you some of the stuff you're looking for. In the series data, like here, we have the sample number, and sometimes the whole data comes with only this, and you're like, oh, is that the control? Is that the healthy, or is that the disease, or is that stage, if this stage of the disease or whatever? So you're like, oh, it says it. If you keep reading, it will have a list of it, and you'll know what you're looking for. If you do need, I've never had to contact anyone because I just haven't. I probably had needed to, but I just didn't. I just used what I had. So here we have it in order, and it says that there are 117 columns. So this is the series data that it comes with. However, it does not have the gene expression data. This is the information of what it is. So here we have a control underscore rep one. That's the repeat one. It's a control sample. They took the data and then they took more of it, and that's the repeat, the second repeat, doing the same process. And it will basically, so you can match this up with that. Oh, wow. Okay. And then it'll have it for all these. You can see the controls, and then it will go into the language. And then, okay, so why do they have these genes? Like, why do we care for the up or down regulation? So if we know what genes are impacted, we can basically reproduce those genes. And they have a way of knocking out a certain nucleotide or gene so that something becomes more active and agile and takes over, like the whole competitiveness, primary competition, secondary competition. It works in the cellular model too. So then they can make some changes. And then just like you have plasma-rich proteins and injections to inject into your knees, to give you more cartilage to build it, because it gives you the building blocks right there, so the body can actually repair it right there because it has it right there. It doesn't have to wait for fibroblasts to get pushed in from movement, but then also it might not have those genes there, so injecting it with those genes so it can use it. Or they could do it intravenously. You'll see a lot of those infusion places where they do create those genes. They basically create it in, I guess, petri dishes. Same stuff they do in vitro, not in vivo. In vivo is in the body. So that's whenever they test it on mammals like monkeys, dogs, bunnies. In vivo or mouse. They like to use the mice. And then from what I've read from the ones I've been looking at, the dog, yeah. So anyways, in vitro, they'll replicate it. They know what works. They inject it with something they know works. Like they'll see certain lab created substances that they put in there, and it gets more genes. Love it. There's like, yeah, create more of this. This is what we need. Oh, kill off that. That's the one that's causing that problem. Kill that one off, and then we'll make more. So then they package it. Okay, and they have all of these nice, ready to go messenger RNA type products that are ready to make those proteins, or they could just make the proteins right there, create it, and then infuse somebody with it, or inject somebody with it into the tumors or into the adjacent cells, or whatever it is they want to do. So you find these genes, and that's a stepping stone. That's a point in space, because then you're like, yes, that's in common. That's what they have. This is the link, that right there. And then you can create something from that using skills you don't have, because you just send it out and then somebody else makes it. And so you don't really worry about it after that. So then you just find those genes, and sometimes, the data is just messy, or it's just not enough. You need better samples. You always need better samples just to be more sure. And then they just do all those tests and see if it works and try to simulate things. So yeah, I only needed the chemistry one because it gets really, really, you have to really know what you're like, be on point all the time for everything. It is like cooking, but like you on a finer scale for everything. You're literally a slave to that project, and you have to do it just the right time. Timers everywhere. You have to like teeter it. It has to be like, you have to go by color. It has to be in a certain range and all sorts of stuff. So we just find the genes, data science. We look at it, we analyze it. We try to figure out certain, I guess, patterns that we can find from these algorithms. So you'll do cluster analysis. Usually, like the unsupervised is if you don't really know what genes you're working with, because you can have a very large file. This is the data from this one. Okay, so let's go into that. Yeah, I don't remember this one being there. Okay, let's look at that. Platform, gene bank accession ID. Okay, and then we have the ID here. Yeah, that's what we were doing. So we're not going to get very far on this, but I do want to say that when I was looking at this before, I haven't actually looked at this in maybe up to 2 weeks because of my car. This ID right here, this gene bank accession number, those go together in the spot ID. You won't really see that much right there. I actually made notes in this document. It looks like the spot ID is fake, and it looks like this because it is a comma separated value, this system, that it's wrong, but it's actually right because it matches up with the row and column of a separate file. They like to piece these together. And so you'll scroll down really far, and you'll see that the spot ID is now showing up. And then this is what's showing up whenever it runs the gene expression. So you'll have this gene bank accession number is supposed to be, wow. Okay, so yeah, those are supposed to be for an actual gene that you can count. Okay, so we're gonna go to, okay, so we looked at this, we went ahead and created the samples to be aligned with the type of sample they are. We have the controls, the language, the mild, the silent. Those are the sample types. Those 4 sample types. That's what we're having. The controls, the language. This was all across the top. Those are those 116 samples, 9, 10, 11, 12, 13, 14, 15, 16. And then let's find out how many of each. 29 healthy, 31 language, 26 mild, 30 savant type, ASD, autism spectrum disorder. Okay, and then I left notes here. We don't have our genes yet, but we do have a folder for it. Let's go into the folder and open up the file one by one. Path. Files unzipped, no folders. That's my folder right in here. So list.files, those are the files. Trying to figure out how to get those in. From what I told you when I unzipped them, and then I took each file out of the unzipped and put them in a separate folder, but kept them in their folders in the unzip because sometimes there's programs that can open it up with that method because it's already been made. Okay, so we took the first sample out of that folder, and then I had 41,472 rows, 18 columns, and it is this one. File 1. And this is what we're looking at. Okay, so whenever we look, the quality control score, that I believe is what the actual score is of the A and the B, which are the, I left notes, channel 1, channel 2, okay, and then unique identifier. It looks like a row number, but actually that is mapped to another file. This other file is actually the platform ID, this unique ID. It's going to be that row. Because if we go to the, hold on, this has 41,472 entries. Look, it's the row in the column RNC, 384, 108, 41,472. And then if we go to this row around 41,472 up higher, all right. Okay, no, we're in the 396. Okay, we need to be in the 41. Okay. Okay, so we're in the 20. So we need to be in 41. 472. Okay. We're getting there. Let's talk about those. 41. Yeah, it matches it to a platform ID because they use these to compare from a standard set. These platforms are created by a bunch of researchers who looked at the genes, and when they were developing this project, came up with it, and they're always altering it. 41, 472. Okay, here we are. 41, 472. Now look at that. It says 384, 108. 384, 108. So the row and column is the this is what we uploaded, the ID column. Now the gene bank accession number doesn't have anything, but now we see here, we scroll down from right there, the start of it, it will tell you the lymphoblastoid cell line, autistic twin. So this is new. Okay, so it has a channel, and then we go into more scrolling. ID ref 41518. I guess that was the only series for that one. That was the platform basically that they used. That's beyond the number of rows in our file. 41472 was the end of it. So that was the platform matching it to that. So it's up to that number. It starts at 1 to the platform. I guess it's the beginning of that platform. Right there, 1 and 1. Also, it does depend on how you read it. 1 and 1. All right, so we're going to say the quality control score is what they use from the quality control of A and the quality control of B, which are two angles. I guess intensities whenever the machine's going through it to analyze for the nucleotide sequences, what the intensity of the cell was in the array data, which can be hundreds of thousands of cells in a microarray sheet. Because, just imagine. And it's meticulously counted that way. And because our cells are so tiny and the nucleus is so tiny. So you can't imagine it physically, but that's what it is. I have never seen it, but I've read it. So that's what it is. That's what they do in the lab. Now, then we go to the second page. Okay, so we're looking at this as well from inside the viewer. Okay, and then this is MR sub-row, sub-column, specific row column in the block, SR and SC, like whenever you see those on the column names, SRSC, those are the spot area, spot row, and spot column. So that's that array with each spot. UID unique identifier, intensity is 1A and 1B for channel 1 and channel 2, right here and here. Row and column, meta row, meta column, block row and column. All right, and then we go into more information for SAS of the flag A, flag B. Saturation of the pixels, saturated, okay, not saturated. And then the actual spot in channel A, saturation factor of channel A, so SA, SF. Quality score is a cumulative quality control score. QCA and QCB is the quality control score in channel A and B respectively. That's in like machine terminology. This uses it's a 2009 publication, so Tiger could have been like a prospective system to really do some wonders, but I haven't seen it since in the later ones. It's usually Affymetrix or Illumina, something similar along that line. So this one was probably an older one. Or maybe it was just so expensive, or maybe it broke down or something. But anyhow, this is what they did. I'm not really sure. Didn't look into that. That's a separate thing. And then it has information. Yeah. So then, reading in the table, this was the platform that we just looked at, the GPL, Gene Platform, PL platform. Okay. Information on that. I don't really. This is what we have. We've just looked at that. It's a platform that many series studies or the series share for their sample information, the GSM samples. They use that in reference. It's basically like the translator code, something you can map to that machine, however that machine works. Okay, and then just going through, looking at more things. Let's go back to the series. Okay, the two. Yeah, so we just need to look up more information. And then we'll figure out what to do. Read that table. Maybe, okay, so I read in another one. This is file 2. Basically, it's the sample 2. The sample, GSM sample, which is the control sample. The second control sample, which is the healthy one. And then reading in the information. So whenever I get data, it's usually already divided into the gene expression value, which I'm assuming is this. And it's for the gene, but depending on how they got the gene. And so for us, our unique identifier, we'd have to map it to the platform that would give the gene. But in our platform, it gives the gene bank accession number. And when you look these up, it's not really a gene, it's a location on the chromosome where you can find other genes. See, let's look it up. We'll just copy that R39069 and we'll go to the internet and we'll look at NCBI gene research. We'll go right there and we'll just put it in there. It's going to show you a gene bank accession number. It's going to be a report of different things that you said. We will go to the genes, gene geoprofiles. Here we go. Here's an accession number right here. And then it goes into a human PIP 5K1B bone marrow stromal cell lines. It references it here. And then if you click on that, that's a separate link to its actual accession ID, which is locus, which is the part of the chromosome where it tells you it's 266 base pairs long, messenger RNA. It's linear. And then it's not a gene. It is a locus where genes can be. So we have to find out what genes are there. And you can actually go in with okay, so blast A will show you the chromosome origin. Here we go. So we will see if we can copy this. Well, we don't want the numbers there. So what happens is okay, let's copy this. We'll go to the notepad. And we'll just put a new one because my computer likes to save all the old stuff. And we will erase that. Erase the row numbers. We'll see if we can just copy this like that. And if Blast A will recognize it. Is it Blast A? I think it's Blast Bioinformatics, basic local alignment search tool. We will do a nucleotide blast and we will put it here. And we'll see if it will find this locus gene. So we are looking, because we see T, it is DNA. Where it is, it'll just look it up on its own. I thought maybe there might be RNA standard database. Maybe it's genomic. Oh, okay. Okay, let's see if it's okay. Database, human gene and transit database. Maybe that's not sure. Show results in new. Okay. Show results in new window. Okay. So, it's gonna give you a list of possible genomic sequences. Okay, so I would get, these are the predicted. I want actual and -1. Now we can go to the graphics of it, and it is going to have this here. It's going to show you the chromosome. It is, here's our chromosome. This is our viewing window right here. So let's zoom out and we will see where we are. This is basically the chromosome. So upstream is going to be like if you ever see a positive on the gene strand, or a negative. So if it's facing this way, it's downstream. If it's facing left, it's upstream. This is an upstream query. This might be the query that we just gave. Let's look. NC 00009. It's been a while since I looked at this guy. Here we are. Okay. So this right here is a gene. It's called PIP5K1B. I believe that is the name of that gene that referenced that gene bank accession number. So here's a gene that's in our window. This is the locus of the chromosome. Now this one, it will tell you when you hover over it, it is, okay, it doesn't say up or downstream, but sometimes it'll do that. It'll show you the gene position and then the gene name, the location, how long the nucleotides are. Remember, they don't use all of them though. This is straight off the chromosome. The chromosome, the DNA of the chromosome. So a lot of these are introns, extrons. Some are going to get dropped because it's part of the protection of the actual gene. And then we can zoom out more and see where we're at. Okay. Here's one. It's going upstream. It's also complementary. PRKACG. So you would have to manually go into each of these once you find the data. You have to arrange the data first to get it into the format you want, but you have to get the gene expression value for this one first. Put onto a big, nice database of basically samples on the top and underneath each sample, all of the genes they have, which we have 41,470 gene bank accession numbers, which we'll say are locus where more genes can be. And then you would just get all the samples there, and then you group them, and then it would go across each row where a gene is across all samples. You would create the row of those samples in that group, get the average, compare to the average of the pathological group or another different group. You would get the gene expression full change values, and you would see if there were changes upregulated or stimulated, they call it, or downregulated or inhibited, they call it. Okay. So, yeah, that's where we're at. Yeah, we've been there for like 2 weeks. Yeah. So, the thing is, I would have to actually, for each of these files, just column bind them. Each sample going down, 41,000 gene accession numbers. I would take only that column of, I would believe it would be I would say the quality score. But I mean, I don't know. Is it? I'm assuming it is because it looks like the average of A and B is the score. So we're going to say maybe that's the gene expression value. Let's see. Well, actually, I've never used the average, so I'll just do 0.2776 plus 0.3926 divided by 2. Oops. You go up. Okay. And then n divided by 2. 0.33. Yeah, so that's the average. The quality control score right here is the average of the intensity of A plus and the intensity of B. So then you get this for that gene bank accession number, which is this that you have to match to the platform, because we're looking at the file and then you match that to the platform by its ID. Yeah. Unique ID. Yeah, so it's in the platform. We just basically go by the row number. That's it. And then these are the row and column. And that's how you know if you're in the right one, because this row and column should match that row number of the platform. Okay, so we would just do that. So we would just look at the column names again of the file that we uploaded. So we would just upload every one of those 116 files. And then basically, we should just, we could do that by the sample number, just do it all over again so that we know, because we'd have to just enter in each one of these and do that. Unless there's a YouTube video on there that will do that. One wonders. It probably is, but I don't know. So that's it. That's what I'm going to be doing. Yeah, I'll just be looking at each one of those, and then we will just keep the so they're all pretty much going. Oh yeah, because then we have to match on each too. Okay. Yeah. So if we keep the quality control score, we have that, and then we do know that the user ID is the row number of the platform. Whenever we'll just keep the user ID, and then we'll keep one file. Well, we'll just keep the row and column number and then the quality control score. So that's the first, third, fourth, fifth, and then the 12th, 13th, 14th. So All right. We will do that. So for file 1, we'll just say, let's look at it again. Okay, so the columns, okay, columns we want to keep are 1, 2, 3, 4, 5, 12, 13, 14. Okay, and then we will column find the other samples of 116 samples total. So 115 other samples. We will only combine the QC score for 14th column. Okay, I'll drink some water. Doing something different. All right, and then let's go ahead and do that. All right, so let's just go back to the file. Unzip the MIDI files. Zip to new folder. Okay, so we saved that as path. We're just going to have to in each one. We're just gonna for file 1 and 2, we'll keep them there, but we're actually going to have a huge table of these. So we're gonna have an environment full of these objects that are going to be by the sample name, and if we keep them in order, we will know. Okay, so in the folder though, it doesn't keep them in order. All right, that's fine. We'll just do a whole bunch of copy and paste though when we do this, so that we can just run each chunk. We don't have to like type in each one, but we do have to replace the file. So let's go ahead and look again at which. Okay, so the sample. Okay, we'll look at that later whenever we combine it. But so we'll go ahead. Okay, so read the table. Comments are that. All right. So we'll read in the first file. We'll keep these we'll keep the columns that we just said. 1, 4, 5, and 14. Okay, so the first file is GSM. Okay, so I kind of want to just keep file, then I have to like match that one also. So we'll just do that. 6518 3 dots. CS is what I used. Because table, CSV, read table. All right, read table. Okay. Read table. Oh, we got to set the working directory too. Set working directory to path. And then we look and we just add an MEV to it. GSM 386.518.MEV header equals true, comment dot char equals the hashtag, because in that file, if you open it up and Notepad lets you look at it, it might look, oh, it's too big. Okay, so like if I go in here and I go here and I look at it in Notepad, this little comment tag in the text tab delimited file, which is why we're using read.table. It is just for commenting. So you can see what's there, the machine used, how you can understand it basically. And then there's our table. After the comments, the table begins. And that's our ID road number that matches to the platform. So you have to look at the file in the text sometimes to read it in. Oh, yeah. So I did read it in the console. So we're going to just read it in. And we can ignore the warning too. And then just copy this. We'll just read it again because it's already named that. So we'll just take the other one and we'll rename it. So it is what we want. That's our first file. And we have a list of it. So basically these are in order. We could just keep doing this from, we'll just have to go down the line. So we go from 518. It's 386. You can see it's all 386. And then we're going all the way to 633. So we'll read it in. And if there is an error in that, then it won't have a file. Then we just can't upload it. So we'll see. Because let's see. 633 minus 518, 115. Yeah, so including itself, pigeonhole theory or something like that. Mathematical reasoning or mathematical. Yeah, this stepping stone to math analysis. So yeah, there you go. We will just do that, and we'll see how long it takes. Keep track of time. I'm getting paid 1 cent per hour. I'm just kidding. I'm not getting paid anything. Okay. So just copy and paste. And then we will be combining these with that as well. But then we will just add it as a vector so that we don't have to do chunks of it. We can just type it in. All of 160 names. Because we don't have a program right away available that we are aware of. Okay, so 1, 2, we're doing this until we get to 116. 1, 2, 3. Oh, you know what? I need to just copy it here. Control C. Okay, so that was 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. 1, 2, 3, 4, 5, 6, 7, 8, 9, 30. 1, 2, 3, 4, 5, 6, 7, 8, 9, 40. 1, 2, 3, 4, 5, 6, 7, 8, 9, 50. 1, 2, 3, 4, 5, 6, 7, 8, 9, 60. 1, 2, 3, 4, 5, 6, 7, 8, 9, 70. 1, 2, 3, 4, 5, 6, 7, 8, 9, 80. 1, 2, 3, 4, 5, 6, 7, 8, 9, 90. 1, 2, 3, 4, 5, 6, 7, 8, 9, 100. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Ooh, I missed 14, 15, 16. Okay. No worries, because we're just going to scroll on up to where we were. And, yeah, so just start going in order. If you need a break, go ahead and take one. So that is 19. Okay, and then we just go right down. 20. Let's save this. 20. Okay. 21. 22. All right. 23. 24. 25. 26. 27. 28. 29. 30. 31. 32. 33. 34. Oh, man, I should make this less bright. Oh, okay. 35. Oh, gosh, I need a little bit brighter though. 36. 37. 'Bingo! Just kidding. 38. 39. 40. Oh no. Okay. 41. 42. Okay, this is 42. 43. 44. Look, it's only been 5 minutes. Okay. Clock those hours in. Okay. 45. Oh, God. 46. 47. 48. 49. 50. 51. 52. All right. 53. Yeah, so you can imagine doing this with each of the files as well. Oh, see, there we go. 53. I'll just say it out loud. 54. If you have ADHD, this is really difficult for you. 55. 56. 57. 58. 59. 60. 61. Oh, you can go to my RPub site and get this file too. I totally forgot to tell you that. Aw, so sorry. All right, so rpubs.com/Janice Harris, I believe. If not, then it's Janice Corona. I forget which one. It shows me as Janice Corona, so I think it's Janice Harris. But no, I think it is Janice Corona. Yeah, Janice Corona. That was my married name. But I also live in Corona, so it could still be Janice Corona. So there you go. 61, 62. If you want to take off from where we started. It's not the last file, but the one before that. It should be anyways, from this date. Date's in the bottom. To learn, whatever that is. No. Okay, so 62. Good. Make sure we're not giving the wrong file name to the wrong file. Okay. 63. Almost there, guys. 64. At some point in time, there will be some people. I don't care. 64. Because I know there will be. 65. All right. And then we have 66. 67. 68. 69. 70. 71. 72. 72. 73. Oh, oops. 74. 75. 78. Almost there. Don't get so strong, soundboard. Okay. 79. 80. 81. 82. 83. 84. 85. 86. 87. Oh, for you. 88. 89. 90. 91. 90. Oh, darn it. I have a touch screen. Accidentally touched the screen. 92. Oh, okay. 93. 94. 95. 96. 97. 98. 99. 2K 600. If you don't get that, in the year 2000, the computers weren't updated to handle a new century. So they thought everything was going to crash and the economy would crash with it. But they actually figured it out and a bunch of people made lots of money. 601. 601. Okie doke. 602. 602. 603. 604. 605. 606. 607. 608. 609. 610. 611. 612. 613. Oh, shoot. 614. 615. 616. 617. 618. We've done 100 now. 101. Only a few more to go. No worries. 619. 620. Okay. 621. Oh, okay. Oh. 622. 623. 624. 625. 626. 627. 628. 629. 630. 631. 632. 633. 634. 635. 'All right. So we need to go look at what our last file is because I don't recall. I think it's 636. 633. Hold on. 31, 32, 33. Wait, did we do more? Hold on. 21. Okay. All right, so 33. Where did we go? I feel like we did 35. Did we skip some? 33. Okay. All right. Those waste those ones. Okay. So that's weird. Sometimes that happens. Compare them. Okay, so we're gonna read in each of these. We could just run all the chunks from the last one we ran. We're gonna have a huge environment of these objects. But what we're gonna do is, so when we read them in, we're only gonna add the quality control number, which is I think it's the 14th column. So we'll just add the brackets, comma, 14 to it at the end of each name. It's going to be a copy and paste. And once we read this in, let's go up all the way to the top. I look at my stats. All right. So 12, 13, 14. So it is the 14th column. We're going to actually copy and paste this because we already have the 18th one. We already read in that one. That's our first file. From that one, let's look at it again. Okay, so we have everything. Okay, so these are all uniform. They're going to all have the same 41,472 rows. We're just going to keep the columns that we said, which is this one, the user ID, and the quality control score, but we're also going to keep the row and column right here, which is that identifies it in the platform. UID in the platform is the row number. Row and column is the number, comma, number in the platform. Let me see it so you can just verify. All right, so we have this one. We're going to go ahead and just add a line here. We're just going to call, let's go ahead and just call it DSM 386518, and we'll just call it start. And it's going to be only those columns that we just wanted. So you do a bracket sign. We want all the rows, but we only want a vector C parentheses of the numbers that we just listed off there. The first one, the fourth one, the fifth one, the 14th one. So we can just go ahead and select that. We can run that line. And then we can go to it and see if it did what we wanted. And it did. And so from here, we're going to add the QC score of everyone. It's going to give a number to each one. So this is the initial one. And so we have to read them in first. But that is going to be the one that says start. That's going to be the one we add them all to. So, I mean, that was like 25 minutes of doing that other stuff. It's part of the data manipulation or extraction. So we got that going. Yeah, we're manipulating the data to get it into what we want. From here, we verified it as we went along. We're just going to run all the chunks. And if there's a problem, run all chunks below. We're going to know, because this is not going to look so great. Oh, yes, so we ran into a problem. Let's see. Where's our little problem at? Okay. 3d6. Oh, geez, that sucks. But see, it flagged it for us. So, can I open the connection? GSM 386. It's not even the right number. So, good thing for that. 386. 532. Oh, dang. That's too bad. Okay. So we'll run that. Now we're going to run all the chunks below. We can save it too, because it's work. That's 30 minutes of work right there. I'm going to lose that. There's lots of time, so I can't do anything. We see all of these objects. These are our objects. They have all of the rows right now, but we are going to copy and paste and add that into its own little table. Yes, I will not leave you until we do that. All right, so we're just gonna call this big table. We'll just call it big. And then from here, we are going to Columbine, yeah, C-bind. Okay, we're going to do the GSM. Where is the start one at? Okay, GSM 386 518 start. And then to this, we are going to copy and paste. Oh, shoot, really? Oh, that sucks. Okay. I thought I copy and pasted that last one over there. So we're going to go and do that. Hold on. We'll just scroll up to the top. A little faster, even. Look at that. Okay. So I guess I didn't copy and paste those. We don't want the 518 because we already have that. That's our start one. Control C in my PC. Copy. Now we paste this. And yes, there are errors because we need to put commas between each one. And, so let's go ahead and we can actually, let's just delete all that. You don't need that. That's not part of the code. It's just from the copy and paste. And we're not going to use the MEV, but we'll just do a substitute from inside Rstudio to substitute that portion of the name. Oh, yeah, we also don't need the actual quotations either. Okay. All right, so we're going to copy this. And then we're going to go up here to this magnifying glass. In the selection, make sure that's selected. We are going to substitute this for nothing and replace it all. Okay, now we're going to go ahead and highlight it again. Because these are objects, we're not, those are objects, there's no quotation on objects. And then we're going to make sure this is selected in the selection. I'm sure you're right there too. And then we're going to put in dot MEV and replace with nothing. Make sure it's selected. That box has to be selected, or else it will go through everything and find and replace it. Let's save this too, because just in case we can close it and open it if there's a mistake. Okay, great. Now, what we can do is make a space, one space. It might put multiple commas though if there's multiple spaces here. Okay. I haven't done this before, not this way. Hold on. It jumps. Okay. Replace with a comma. And it's still selected. All right. Awesome. Great. Now there's a double comma here. Don't want that one. Oh yeah, so right here too. Ba-dee-ba-dee. There's another double comma. Yeah, no comma here. And then right here. Okay, so now what we want to do is see if it will do. Okay, let's save this. In the selection. Now we want to replace the comma with bracket, comma, 14, comma, in selection selected, and then replace it. Oh, that sucks. It didn't replace it. I don't know why it didn't. That is so weird. Okay, comma, replace. Huh, that's very weird. Hold on. In selection. Because there was a space there. And now we'll do it. Okay. Let's make sure because we are adding only the 14th column. Correct the little sorry. No, oh yeah. Now we need to add a comma actually to the end of. We need to add a comma to the brackets. the ending bracket. Ending bracket, and with a bracket, comma. All right. Now, okay, so I'm confused. Make sure that this is the same. Okay, so it's not binding. Delete. Okay. And then delete that. I have to make a few little changes. But I mean, I think it's slower than manually doing it. Okay. So jolly, so jolly. That's why I have dogs. Alright. It's easy to get lost doing this stuff, okay. Okay. Okay, and then we'll add one here. And this should create our table that we want. Don't see any. Yeah, okay. So we have that. Okay. So let's just run it. Oh, something's going on. GSM 36576 not found. Well, that's too bad. Must have skipped a number. Let's go up 576. 76 is not there. See, isn't that great? Wow, that's crazy. I skipped 76 and 77. Okay. $76. That is weird, but okay. Sometimes, it's easy to get lost in track, but thankfully Rstudio is there. So we caught an error. Now we're just gonna run these. They would have probably given me the same error for 77 because I did have 2 extra ones afterwards and we did count that one. So at least we did count that right. Okay, so let's just run this again. Okay. Now there are 119 variables though. So there should only be 117. Great, let's just go look. Oh yeah, actually there should be more because we have these other ones too. So 116 plus 17, 18, 19, 20. Because there's 117. Wait, hold on, let's look at the data frame. 116 samples. 116 samples, but we have 119 columns. So 116, 17, 18, 19, 20. We should have 120 columns. So we do have a column here that is an extra column. We'll go ahead and look at that one. So this first one is actually for GSM 386.518. For the column names, let's go ahead and remove that 14 out of it. This is the quality control score, but we can remove that in the column name. Okay, so column names. Let's just look at the column names of our data set. Hold on, let me see that. I'm not sure what. Oh, that's when I copied and pasted. What? Where's the little arrows at? Yeah. All right. We'll just run the line. We'll have to go through each one and see which one is stupid, or we can scroll up. All right, but for now, let's get rid of the. Okay, let's change the first, second, third, fourth one. The name to GSM 386.518. And then let's get rid of the column names of big G sub, the bracket comma 14 brackets column names big. Our big data of all these samples and the quality control score. And I think we need to use fixed equals true. Okay, yes, because we didn't put anything here, so we're just going, because we don't want anything there, so we'll just run it again. It's good that it lets you know. Now let's look at our column names of our big data. It didn't replace it. Huh, that's weird. Column names, I should have replaced it. All right, maybe I don't need to add that one. Oh, you know what? There was a space here. That's why. Okay. Ugh. I should have put fixed equals true. And then when this happens, we're going to have to rerun it again. Darn it. We have to run it again from this one because we no longer have the same data that we're working with. And we have to run all the code there so it's the same whenever we run the whole entire script. Yes, there we go. That's what we want. There's 119 columns now. Oh, yes, 119 columns. So 116, 17, 18, 19. Perfect. That's what we want. Great. So we have our 116 samples plus the row R in column C and the UID for unique ID that matches row number in platform GPL table to the gene bank accession number. Yes. Now what we're going to do is take a little break because I have to go to the restroom. Okay, we're back. All right, let's have some water. 9:20. Started at 7. Okay, and now I'm going to take you through. These are the quality control scores. Let's go ahead and get that. We didn't page table. That's the R Markdown library that we're using to put a nice data table of what we just created in the Nicher program when it runs it and prints it to the archives. Okay, so those are our rows, our columns. Okay, now what we're going to do is we're going to look back again at that series data. I think it was series 9. Okay, let's go back to series 9. And we'll look at that. So we can see where we need to match what we can call our control group. Back started hurting. Oh, right where my gallbladder liver. Okay. So, we have the controls in it, and then for each of these samples, we can basically just match it to our samples. Now, we could just column all that, or we could just list which numbers they are. So if we look back at the first control, which we're going to call our healthy. So the controls for healthy start with GSM 3D6 518 through. Okay, let's just keep looking at this. GSM 386 546. Okay. Let's just make this really kind of a little bit better. And then the language is GSM 386 by 47 through okay. Okay, so it is at GSM 386 577. GSM 386 577. And mild is GSM 386 578 through 578. Oh, we went too fast. Okay, GSM 386 603. GSM 386 603. Oh. We did actually have a class. We already have the class vector for all of these because they are in order. So we're going to use that later when we make our matrix. Because we have the samples right now. But when we get our top genes, we'll make the matrix, and that will be our class feature. Because the top genes will be reversed or transposed data frame, and the samples will be all 116 as samples on the rows. Right now, they are the headers. And we're looking at all the genes, which is like 41,000 genes. So we can't possibly, I mean, if you do PCA analysis, it can do it, but it's a lot of work. So it's just easier to filter out with this full gene. They also have programs for it. I haven't used them in so long, though, because I just prefer this way. Okay. And then the savants are GSM 386 604 through GSM 386 633. All right, so what we're going to do is we're going to add some more columns or features. Feature columns, you can call them fields. Some people call them fields. I used to call them fields, but then someone said, oh, that's only if it's like access or an access database or Excel. So like no one calls it fields. Or maybe SQL, I don't know. Which is everyone uses SQL in the big computing areas. So, okay, we're going to add these columns. We're going to name them by the mean value of that group. And we have 4 groups. So we have the controls, which are the healthy, we have the language types of autistic spectrum disorder, the mild types, and the savant types. We're going to have a class feature that we made ahead of time. But again, you can get this document and do all this stuff to it. I'm going to also share this document because I'm going to publish it to RPubs after we do this stuff to it. So, and then you can just download it from there. And then you can get your R and your Rstudio and run it. And so you can start learning. I think this is a great tool for that. All right, so let's go ahead and add those columns. We're going to do the mean. Pretty simple. There's actually just a base function for that. To do the median, though, you have to actually do a little bit more, and I don't remember it, so I have to revert. I have to look at one of my other documents for that. So we're just going to add the averages and then get the full change over the healthy or the controls for each one of these. Okay, so big by healthy mean, the average. We're just going to do row means big vector C. Oh, shoot. We don't have the actual column number, though. Oh, wait. We do, though, because it's 518 through 546. And then we go... We're going to have to just do that in this control panel right here. Column names of big. Okay. So you can see right here, we already know which columns are the healthy, 518 to 546, but we have to go down here. So it's actually the first, second, third, fourth one. And you just put colon because it's in order through 546, which is right here. And that one is the 28th, 29th, 30th, 31st, 32nd. So the 4th through 32nd column. Then you're going to go big by language mean. Row means big. C. 33 to, because it's the next one in order. Language is 547, which is, let's start 28, 29, 30, 31, 32, 33 through 577, which is right here, which is the 63rd column, one before 64, so 63rd column. Then we're going to go add a column that says the mild mean. '64 through wherever 603 is. Oh, okay. This one right here. And that one is the 82nd, 3rd, 4th, 5th, 6th, 7th, 8th, 9th. The 89th one. Then we're going to go and add one for the savant mean. It's going to be the 90th through the 119th column, because that's the last one, which is 633, 604. Let's just verify. 90th. Yes, it is. One before the 91st one. Okay, so let's go ahead and run that. And then we'll go ahead and look at our column names of this. And we should have those columns at the end. Those are our mean values of that group. And then we can look at it. Let's go ahead and get rid of these other ones that we're not really using. We'll just keep that one because we're going to use that later. Probably. Maybe not. I don't know. Yes, we will, because we need to get that gene bank accession number when we get our field changes. Okay, so there are 123 total columns now, and then there's the 50, so we have to go to the end. Scroll along to the end. And we have our mean value of the savants for a gene expression value for all 41,472 genes. The mild mean, the language mean, the health mean. Now, we're going to take the fold change of each of these types of autism spectrum disorder over the healthy mean. So we'll have a language fold change over healthy, mild over healthy fold change, and a savant over healthy fold change. And then we'll go ahead and order it by each one. Take the top 10 over and under expressed. Or we could just save that for another time. And we'll publish it. And then, actually, let's do that. Let's save it for another time, and then we'll publish it. But let's do the fold change first, just so we know, and we'll order it later. Because I'm not sure, this database might be too small to share on RPubs. I mean, not on RPubs. Yeah, for sure. Yeah, I can't show. So I share it on Google Sheets, and then sometimes it's too big for that. And then I have used Kaggle for the really big ones, data tables. And we're going to go ahead and add the full change. Let's do it. Big by language. We're just going to call it language healthy FC. Let's call it FC, language healthy. Big by language mean divided by big by healthy mean. Okay, that's it. Big by FC mild healthy. Big by mild mean divided by big by healthy mean. Big by FC savant healthy. Big by savant mean divided by big by healthy mean. Page table. Big. So we'll just scroll on down to all of these columns to the end, because that's what happens when you're in the page to view inside. And we can see our columns, and these are the full changes. We can do a summary stats of these ones on the last one. So big, and then we'll just do columns 1, 4, 5, 6. 124 to 126. Those are the full change values. And we got 0 to 12. There's no NAs, so that's good. No infinities, that's great. So we can basically use this, but let's get rid of the zeros. Because when you order it and there's all those zeros there, like, let's just order it by language healthy. You'll see. Big language order. Big order. Big by language or FC language. Descending equals true. And then we'll just look at the tail of big language order. Which is like, Oh yeah, big by, by tail, big language order by FC language healthy. Big by FC language healthy. Argument lengths differ. Big language order. Okay, so that's the data table. Order. Yeah, I'm not sure what's going on here. That's weird. Descending equals true. Argument lengths differ. All right. Sometimes stuff like that happens. Order. Okay, so that's the name of the table. Order is the function. That is the name of the variable. Okay, let's do. Oh, no, I got that in there too. Order. Huh. Okay, I don't know. It usually works, no problem. Argument lengths differ. It's in this first one right here. Big ones order. Okay, let's just look at this. Because you can actually go to the full change. and order it. Oh, there's only a few that are 0. There's actually not that many. Order. Okay. Let's see on here in the mild. Oh, lots of 0s in the mild. We'll be getting rid of those ones in that database before we take the bottom 10. And then the solid. Okay, so there's no 0s. Looks like. So the mild, mini 0s. I've seen which is only a few. Okay. So, not really sure why that's not working. I usually don't have a problem with that working. Let's just look. Order. Okay. Decreasing. Oh, it's decreasing, not descending. Decreasing. And that's to tell the last 4 zeros. Okay, let's go ahead and make a table of the top 10 and the bottom 10, but we're going to first make this so that it's larger than 0. So big. Language. FC. Let's see. Okay. Okay, top. Let's do top. Oh, wait. Okay, big. We'll do this first. These are just placeholders. Big language order. And then we're going to big language order. Okay. Big. I think we can do this way. Order by FC language healthy greater than 0. This should do it. I mean, if not, I'll have to do a different one. Okay, and we can see that there are 4 less because there were 4 zeros in there. 4 less observations than the actual data. So it has filtered it. Now we're going to take the top of those because the dimensions are, we're commenting this, 41, 467 by 126. Okay, so we're going to now do the top language, FC. We're going to do the top 10 upregulated, stimulated, and the top 10 downregulated, inhibited. So it's basically just a vector 1 through 10 for the first one, and then that's going to be the last 10, which is 41, 460, 58 to 41, 58, 41, 467. Make sure the comma, that's to separate the rows from the columns. And this should be 20 observations long. Let's go ahead and do that. We can just run the whole thing again. Sorry. Top language is 20 observations. Okay, those are our top genes for language. Now we're going to just look at them. We'll put it in the document. We'll just run this line. And so now we have it, all of the data, but the top genes, and it goes, I did include all the samples, so you can see all the samples, but we're going to look at the fold change for language at the end, and these are going to be the top, the values, so it is the ordered in descending order, the top overstimulated, over stimulated, upregulated, and then the down regulated, inhibited. So then to get the gene accession number, we'd have to look at the user ID. And whenever we match the row number to the platform row number, we will see that the row and the column number should match. So 8750 in the platform should say 82, 2 in the platform. And then we will take that gene bank accession number. So what row is it? 8750. We're going to go ahead and scroll to 8750. Oh, went up. No, we did. We did. Okay. So, slightly. Okay. From here. Almost there. Oh, this is great. $82 and 2. As you can see right here, 82 and 2. This is our gene bank accession number. This is basically our key. So 8750, 82 2. Let's look and see. Oh, what just happened here? Hold on. That's the wrong one. We're looking at here. 8750, 82 2. True. This is true. This is true. So this is one of our top genes. That's one of the overstimulated or the upregulated, stimulated. And then we can just assume the rest are the same. So now we're going to just do this for the mild full change, and then also the mild has a lot of zeros, so we have to filter it first. So we'll just copy this and just substitute language for mild. Mild. Order. Okay, we didn't filter out the zeros yet. Okay, so it's weird. Is this? Oh, okay. So now we're going to filter out the zeros, copy and paste. Just shoot. Darn it. Okay. No worries. Sometimes that happens. Instead of pressing Control C, I press Control V, and I pasted the last thing. So I can't go back, but we have a history right here, and it was only a few codes earlier. So, yeah, we actually don't need this one that I just copied and pasted. What we do need, though, is the one that filtered it out. Order. Okay, so this isn't all of them. Oh, this is it. Right here, we're going to add that to our source right there. But we're going to also have to put the tilde, or that's not the tilde. It's like the, I don't know what that's called. An apostrophe. It's right by the tilde sign though. Hold on. Shift. Okay, for the curly brackets, and then the same thing. All right, and then right after this code, there was this one. Let's see. 0. Oh, because we ran it again. All right. I mean, we don't really even have to control. Okay, we need to put that little R in there too. Mild. Order, big order. So actually, this doesn't even go. Okay, so this was what we wanted. Okay, order, decreasing the true. and then we change the name to this. So there was that other code that needs to go to the source, because this is a script, and after we run it, everything should be the same. When we change one little thing because of an error, we're like, oops, let's go back, blah, 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 and you don't change it in the script, whenever it gets ran again, there's going to be a bunch of problems. That could just be one of them. It could just be one problem, but it could be a whole bunch of problems because you keep doing that. So what happens is it just layers it on. It goes down the line. You don't change it. It's kind of, it saves what you do. So if you make a mistake, you can go back. This history is here for you to recall. You have to fix the mistake so that it runs properly in order. Okay, so we're actually copying this, and we're going to substitute the language for mild, because this is where we're getting rid of the 0 entries, because we don't need a bunch of zeros. They don't tell us anything. That's just like a place marker, because we're not getting the top inherited genes. We're just getting the ones that didn't show anything. And they're going to be alphabetized. So, yeah, that's not useful. We'll just manually do this. We don't need to use the search and find. Oh, I don't think the glasses will help me. I do have my reading glasses though, but I don't wear them ever, really. Probably should, but I just do. Okay, so this is our new data. We ordered the data, then we're going to get rid of the zeros, and then we're going to take, so we can't really run this yet because we don't know what the dimensions are. So we're just going to run it line by line. Let's do this one first. And now we know the dimensions. We go back to our environment tab. Right here, big mild order. Hold on. That's weird. Usually it would have, but I was on it. 41,472. We got to run this one first, not the whole chunk, so we can get the new dimensions, because it's getting rid of some zeros. So it's going to be smaller than this. 41,450. Okay. 41,450 for 126 variables. So we want the top 10 and the bottom 10. 41, 4, 41. 41, 441 to 41, 450. And it should be 20. Top mild of these 20 observations. Good. Now let's go ahead and display that. And it will be in order. For the mild, we'll do the savant next. So it's in order. Those are the top stimulated genes, and those are the top inhibited genes in the mild autism spectrum disorder. Keep in mind, reading from the research, 2% of the people classified on the spectrum of autism had genes that they found. So here we go, just to let you know. Sometimes the tool might not be a good indicator, or they might have something else, but it's a start. Well, it's not really a start. It's just like, it's a process. Okay, so we'll go ahead and do the same thing. We'll just be more careful. We'll copy this, but we're gonna Control C and not accidentally Control V. And we're going to replace the mild with savants. Okay. And then for this one, let's just go ahead and use this tool right here in the selection. Make sure that is selected. And then we'll replace it with Savant. Okay. Make sure that's selected because it'll go through the whole document. It's a lot of rules. Okay. Now see, we can't run this line because we need to know after we run this how many observations there are. Well, after we run this line, we'll know how many observations there are. So we'll just select and run this line. And there are 41. Oh yeah, that's right, because there were no zeros. 41,472. There weren't any zeros in the solvent one. So it's going to be 41,463. 41,463 to 41,472. And that should be 20 observation. Good, it is. See, you could tell 20, 20, 20. Good. So we got the top 10 inhibited and stimulated and the top 10 inhibited again. So we'll get rid of that. Now we got the top genes, but not really, not yet, because we have to merge these by, we have to make another row on the platform for the user ID. And with this user ID, we're going to merge by that user ID. And then we're going to get the gene bank accession number. Then from there, we'll go find the genes manually. That would be another time. But for now, we can just show you that. We'll do a few minutes. All right. And we only need the first 41,472 rows of the platform because it is the row number. Okay. Oh, you get lost in this. I do. Okay, I work tomorrow, but not till afternoon, so this is fine. I don't care. We'll just do this one last thing. I'll share it on our pubs, and then we'll get back to work on it later on. Okay, so our platform, we want to make it smaller because it has way too many rows. Look at that, 8 million. Yeah, we only need like the first 42,000, but actually only 41,472. So we're going to call this GPL 41472. Actually, that sounds too much. Okay. We're just going to call this short platform. Okay. Platform. And then we just take the first 41,472 columns and rows and all the columns. And then we just look at the dimensions of the short platform. Okay. Now we want to do short platform. We want to add another another column by UID is the row dot names of short platform. And then we'll go ahead and look at a page table version of it. And see what happens. Yeah, we have the row ID. So now we can merge by this user ID because the ID here is the row and column. And we're going to get that gene bank accession number from each of our top ASD groups. And It's UID in let's get rid of the big one. We don't need that anymore. UID. So it's it's gonna be that. So we're gonna go ahead and top language. Or actually let's just do language. Language 20 GB. GBank. Okay. So now we're going to merge. We're going to go ahead and merge the top language FC and the short platform by.x equals UID by.y. Okay, let's see. Okay. Equals UID. Page table. So now we have the UID and the GeneBank accession number at the back. Here we go. Here's the GeneBank accession number right next to the FCs. And we also have all the samples. So what we're going to do is write this out. Language 20 word.csv language 20 GB to language will change top 20 GeneBank accession.csv row.names equal false. And I'm going to put here eval equals false for when it runs again, because I don't want it to run again just in case there's an error from some mistake later on, because that does happen sometimes. Okay, now we're just going to look in our files and we'll go to modified. And it doesn't show it, but it's there. Oh, actually it's in this one. You can also go to working directory and it should be that one. And you'll see it's right there. There. That's our file to use later. Okay, now let's go ahead and do the same thing with the other ones. Let's just copy that. Let's just copy this whole thing. And mild. Goody. All right. So we have that. Because we don't really need this side right now, what we need is actually the end column. So we don't need to rearrange it. So the user ID is there. We already have the GeneBank accession number because we merged it. And that's what we want, because when we go and look at that chromosome at that GeneBank accession number, some of these don't have anything, but these ones do. And so we can find out what loci of what chromosome that is located at manually, because that's what you're limited to, because that's what I know how to do. But it's an ongoing process. You always learn new things, and people are always putting stuff out there. So I think I can copy and okay. So let me just do the last one. Okay, now we're doing the savant. This is the reason for doing this whole one right here. Okay. Oh, did we write that last one? No, we didn't, did we? Okay. So we'll write out the mile 20 GB and the savant 20 GB in the same one. Just make sure everything looks good. Well, fine. Okay, yeah, we're at our limit. Okay. Okay. Okay, so right. CSV mild 20 TB gives you a list of things. Mild field change top 20 G bank accession.csv name equal false. Right, .csv savant savant fold change top 20 G bank accession.csv name equal Nivell equals false. Okay. That's false. Okay. I'll just reset it by going to the good. They're right there. See. There's a language one right there. Okay. We can actually upload those and share those links. And then we'll publish it to our pubs and end the session. So let's go ahead and language top 20 here. Mile top 20 here. Savant top 20 here. Now we have to Control S save. I'm going to go to my Google. I am here. We're going to go ahead and upload. So we'll upload each of these 3 short files. They're going to be in my desktop under this folder, and it is, we'll do this one first. Import data. And it has our data. Oh, no, I don't want to upgrade. Share. Oh, language. Top 20 ASD GB ACC. Anyone with the link? Copy link. And copy done. Paste. Okay, next one's mile. I like to control this because I do. Sometimes things happen. Okay. All right, and then we're going to upload the miles. Okay. Open that. It saves the name, so I can share it. And you want the link, and copy done. Paste. Control V paste. Control S. Savant. Now we do the savant one. Import data. Open now. Save the name. You can share anyone with the link. Copy link. Copy. Done. Control V paste. Control S there. All right. Also, launch. We got our top 20 things stimulated or upregulated and inhibited or downregulated. We will have to go to another site to get the gene names from the gene bank accession IDs using the last one way of retrieving those genes for our top genes in ASD, autism spectrum disorder. Okay. So I'm just gonna say that and knit it. And you will see eventually something should come up. Let's see if it's knitting it. It could be that I'm sharing. I'm not sure. So weird. I usually don't have this problem, so it usually works fine. I think it's because I'm sharing this. And it would be vital information. So I'm going to go and stop. I'm going to share this to my RPub site, but look up rpubs.com/Janice Corona. Oh, look at that. It says it's doing the job. Oh. It's a little slow though. It's because of the screen sharing. So I have to give my personal information. I'm going to go ahead and stop sharing the screen here. Yeah, my eyes do get tired. That's the thing with data science and beer and dogs, especially after a long day. I got up early, so they get dry and tired. Okay, so you guys know where to go. Hope you enjoyed this whenever you watch it. If you watched it, it's kind of nice to have something to do. I think it keeps you busy. It's like playing a game. And then you know these genes. And then you're like, oh, maybe you solved something. It's rewarding. Because it makes it sound like you did some good in them. Actually, today I was at Aldi's and this lady was paying with cash. And she dropped $100 bill. And I was like, oh my God, that would be so tragic. Because she has a child with her. I would hate to lose money if it just fell out of my wallet. I'm like, here you go. Here's your money. She's like, oh, thank you. That's so nice. I'm not someone who's just going to take money. I actually did do that a long time ago. So what happened was I was at a Rite Aid, I think it was, or maybe it was a Walgreens. And these guys were buying some beer. And it's not like I took the money because it was theirs. It was because it was already on the floor. And I don't know whose money it was. And it was $100. And then what happened was I spent it, and I don't remember on what. It was like, I don't even know. But then it was like the universe gave me bad karma. You know what happened? Some asshole fucking sideswiped my car twice. And then they said that it was my fault. I was the one who did it. So there's a record of that shit. And they arrested me. They arrested me and said I was on something. I don't do drugs. I don't drive or drink and power a motor vehicle. Okay. So, anyways, I was arrested, spent 5 hours in jail, the worst hours of the day because it's the end of the day, and it was until 2 a.m. Plus, they took my fucking sweater away from me, so it was cold, 60 degrees. I was 20 pounds lighter. That fucking asshole, Neff, piece of shit, fucking misogynist. Anyways, if you ever run into him, he's a coward. Anyhow, he's the one who decided it was my fault that happened. So that wasn't universal karma. That was some fucking prick. And that's what happened. So anyways, I don't take any shit that doesn't belong to me. Okay? I'm gonna drink my beer at home. You do you. I do me. Anyways, there is this is what is it called? Fermented. So anything fermented has the gut microbiota for your gut, your enteric nervous system. You guys have a great night. I'll talk to you later at my next data science and beer. I only drink one beer. You know, when it's by myself, I drink like 2. All right, I'm gonna end this. Okay, have a good night. Bye.